Hi there, in this video we are going to discuss apoptosis, its mechanism and various conditions in which apoptosis is induced and some other mechanism of cellular death. Moving on, apoptosis is a pathway of cell death in which cellular activated enzyme degrades cells own nuclear DNA, nucleus and cytoplasmic proteins. So what happens in apoptosis? Cellular digestive enzymes are activated and these enzymes digest cellular own components, but there is no release of enzyme outside the cell. As a result, there is no inflammatory response in apoptosis. So what happens in apoptosis? In apoptosis, plasma membrane of apoptotic cell remains intact. There is no damage to plasma membrane. As a result, there is no leakage of enzyme and there is no inflammation. So significant apoptosis even may remain unnoticed because inflammatory response is not induced. So what are different causes of apoptosis? Apoptosis can be physiological as well as pathological. So here we see the different causes of apoptosis. So in physiological condition, apoptosis may be during the normal developmental process. For example, the interdigital skin or interdigital structures during developmental process are removed via apoptosis. Similarly, apoptosis can also be in hormone responsive tissues. When there is abundant hormone, the hormone responsive tissue grows, but when a hormonal signal is deprived, the cells die due to apoptosis. Similarly, in immune system, when there is an infection or a immune response, leukocytes increase in number, but at the end of immune response or when the infection is over, the such leukocyte die out by apoptosis. Similarly, in autoimmune diseases, the lymphocyte which react the self antigen, recognize self antigen, can also be vanished or destroyed via apoptosis. So when apoptosis occurs in pathological condition, apoptosis is mostly induced when there is a damage to DNA by radiation or cytotoxic drug, or there is accumulation of misfolded protein into the cell, or there are certain viral or bacterial infections. So here in this table, different causes of apoptosis are summarized here, which can be physiological as well as pathological. So physiological apoptosis occur during embryogenesis, and during embryogenesis, apoptosis is mostly due to loss of growth factor signals. Cell apoptosis is basically regulated via combination of two signals that can be growth signal or survival signal and death signals. So if survival signals predominate, cell continues to survive, but if that signal predominates, apoptosis is induced. So basically, a balance between survival signal and death signal regulates the apoptosis. So during embryogenesis, there is loss of growth factor signaling, which is survival signal. When the survival signal is lost, there is apoptosis. Turnover of proliferative tissues. These tissues which are responsive to hormones are normally continue to divide. For example, intestinal tissues. In intestine, the cells at the bottom continue to divide, and cell at the top are lost by apoptosis. And this is also due to loss of growth factor signaling or survival signals. Similarly, hormone-dependent tissue, when a hormonal supply or hormone level declines, these tissues also undergo apoptosis. Similarly, decline in leukocyte number at the end of infection, and this is also due to loss of survival signals. Elimination of potentially harmful self-reactive lymphocyte. This we will see in the lymphocyte development, how tolerance develops, and this tolerance also basically a mechanism of apoptosis. In pathological condition, apoptosis is due to DNA damage, accumulation of misfolded proteins, and certain viral infections. So this is basically activation of certain receptor, which is BH3 receptor, which we study here on, and the mechanism of apoptosis. So when BH3 receptors are activated, then they stimulate the apoptotic pathways, which lead ultimately to cellular death. Similarly, accumulation of misfolded protein also is through BH3 pathway, while viral infection induce both pathways, BH3 pathway as well as the pathway via death receptors. So here we move on towards the mechanism of apoptosis. So in mechanism of apoptosis, it is here written that there is a delicate balance between the survival signal and death signals, and this balance controls whether cell is going to die or going to survive. So there are two pathways through which apoptosis occurs. One is mitochondrial pathway or BH3 pathway, and other is death receptor pathway. So what happens in mitochondrial pathway? In mitochondrial pathway, there is a protein which is cytochrome C. This protein is a component of the electron transport chain, and it also has ability to induce apoptosis. So it is basically a pro-apoptotic protein. So this protein is normally present in the intramitochondrial membrane. Suppose here this is mitochondria. Suppose this is mitochondria, and this is inside matrix, this is inner membrane, this is outer membrane, this is intermembranous space. Here cytochrome C is present here, and it is mainly involved with the electron transport chain which is going on in inner mitochondrial membrane. So this remains here and does not move to cytoplasm. This is suppose cellular cytoplasm. So what happens, this membrane, outer mitochondrial membrane is not permeable to cytochrome C. So, this is cytochrome C protein, and it is entrapped here. But there is specific protein family which is BCL2 family. This BCL2 family, and this is a member of protein family. This BCL2 protein and BCLXL. These are two proteins which regulate the permeability of this outer mitochondrial membrane. How they regulate the outer mitochondrial membrane? Basically, they control two components, BAC and BAX, BAK and BAX. These are two pro-apoptotic proteins. These are pro-apoptotic proteins, and these are anti-apoptotic proteins. So these BCL2 and BCLXL control the activity of BAC and BAX. How they control? These are basically channel protein, and when they dimerize, they produce a channel here. If suppose this is a BAC protein, this is a BAC protein, and this is another BAC protein. So these are not dimerized; they are separate. When they are separate, there is no channel here. And center, let me explain. There is BCL2. Suppose here BCL2 lies and BCL2XL lies, and they prevent the dimerization of these BAC protein. But when the activity of BCL2 and BCLXL decrease, there is dimerization of BAC and BAX. So when they dimerize in this way, and when they dimerize, a channel is produced. Same goes for BAC, same goes for BAX, and they produce a channel due to down regulation of BCL2 and BCLXL, or inhibition of BCL2 BCLXL. This cytochrome C flows out through this channel into the cytoplasm. When it flows out to the cytoplasm, it activates caspases, which are proteolytic enzymes, and when they are activated, they further activate multiple proteolytic enzyme, and when these enzymes are activated, they undergo the breakdown of nucleus and cellular constituents. So in this way, apoptosis is induced. So how, when it is down regulated, when BCL2 and BCLXL are down regulated, these are down regulated when there is a damaging signal or damage signal which is sensed by another protein which is BH3. So summarizing here, there are two proteins, BCL2 and BCLXL. These two proteins are anti-apoptotic protein, and there are the two protein BAC and BAX which are pro-apoptotic proteins. BCL2 and BCLXL normally inhibit the activity of BAC and BAX by preventing their dimerization. There is another protein BH3 which is a damage sensor. When there is cellular damage, BH3 senses it and it inhibits these two anti-apoptotic protein. When these anti-apoptotic proteins are inhibited, there is dimerization of BAC and BAX. When they are dimerized, they produce channel here. When channels are produced, then cytochrome C flows out into the cytoplasm. When cytochrome C flows out into the cytoplasm, it activates the multiple enzyme which catalyze the digestion and breakdown of cellular components. So in this way, mitochondrial pathway works. So all these things are explained here in the text, that BCL2 and BCLXL, these are anti-apoptotic protein, BAC and BAX are apoptotic proteins, and BH3 is the damage sensor, and it activates in the end caspase 9, which further activates multiple protein and enzyme which lead to cellular fragmentation and formation of apoptotic bodies. So here it is same thing is explained here. These are regulated B cell to B cell X cell, and these are back and backs are apoptotic proteins and BH3 sensor, and when damage is here, it senses and it activates their dimerization. Cytochrome C leaks out and it activates the activity of caspases, which lead to activation of endonucleases and enzyme that catalyze the breakdown of cytoskeleton, and ultimately cellular fragmentation occur, and there is formation of apoptotic bodies, and these apoptotic bodies are in the end are phagocytosed by phagocytes. So coming toward the next mechanism, which is the death receptor or extrinsic pathway of apoptosis. This mitochondrial pathway is intrinsic pathway and death receptor pathway is extrinsic pathway. Because in mitochondrial pathway everything is happening inside the cell, but in extrinsic pathway there is some external ligand involved. So this extrinsic pathway basically controlled by tumor necrosis factor receptor, TNF receptors. One of the type of this receptor is here FAS or FAS receptor. This receptor is present on certain cells, and when this cell having FAS receptor binds with the cell having FAS ligand, there is domain of this receptor inside which is called death domain. This death domain interacts with adapter proteins, and when they interact with adapter protein, this protein in turn activate the caspase 8. This caspase 8 in turn activates the endonucleases and the enzymes which digest the cytoskeleton, and ultimately there is formation of apoptotic bodies. So here you can appreciate here, this is a receptor, TNF receptor or FAS receptor. Here comes its ligand. When ligand binds to it, its intracellular domain, which is death domain, interacts with adapter protein. These adapter protein in turn activate caspase, and this caspase in turn lead to activation of endonucleases and multiple enzymes which digest the cellular cytoskeleton and cellular constituent. As a result, lead to the formation of apoptotic bodies. And these apoptotic bodies are formed. These apoptotic bodies in turn lead to the release of eat me signals. They produce several signals which induce the macrophages and other phagocytes to come on and eat them. And these apoptotic bodies have intact plasma membranes, so their internal constituents, whatever is happening inside, does not influence the extracellular environment. So whatever is happening inside, or whatever the constituents are present inside the apoptotic bodies, they are not released into the extracellular space because the plasma membrane of apoptotic bodies is intact. So before the plasma membrane get also damaged and constituents are released out, macrophages come and macrophages eat them. These macrophages are stimulated by a specific protein here, phosphatidylserine. Phosphatidylserine is normally present in the inside of cellular membrane, but in these apoptotic bodies, what happens? This phosphatidylserine moves on from intracellular space to extracellular space. When it moves intracellular side to extracellular side of cell membrane, when it shifts to outer leaflet or extracellular side of membrane, suppose this was cell membrane, normally phosphatidylserine was present inside, but in apoptotic bodies it shifts to outer leaflet. When it shifts to outer leaflet, it attracts macrophages, and these macrophages come and eat these apoptotic bodies. Further, they also release soluble factors which include cytokines and chemicals, chemokines, which attract macrophages and they are eaten upon. So in apoptosis, there is no inflammation induced. When there is no inflammation, it becomes difficult to trace the apoptosis going on. But apoptosis may be visible in case of organ atrophy. When there is massive apoptosis in the organ or tissue, that can be noticed in the form of atrophy. Now we come toward other pathway of cellular death. So other two pathways of cellular death discussed here. One is necroptosis and another is pyroptosis. So necroptosis, as the name indicates, it is combination of basically necrosis and apoptosis. So what happens here in this condition, there are specific receptors which are receptor interacting protein kinases, which are activated, as well as TNF receptors also activated. So second pathway or extrinsic pathway of apoptosis is stimulated, as well as necrosis is stimulated. So there are some features of apoptosis and some features of necrosis. So it commonly occurs in ischemic injury as well as some bacterial viral infections. In this condition, there is also features of apoptosis like formation of apoptotic bodies, but there is also some signs of inflammation. So it is basically a combination of inflammation as well as apoptotic bodies. This is called necroptosis. However, this is topic of detailed research, and it is not known how it plays role in the human pathology. Next is pyroptosis, and pyroptosis, as name indicates, pyresis stands for fever. So it is combination of apoptosis and fever. In this condition, there is a specific cytosol damage sensing receptor in the cell, which is called inflammasome. When this inflammasome is activated, it leads to formation of certain chemokines and cytokines. Some cytokines induce fever, some cytokines recruit the leukocytes as a result, and some of them induce apoptosis. So in this condition, there is inflammation, there is apoptosis, and there is fever. So in necroptosis, there was feature of inflammation and apoptotic bodies are apoptosis. In pyroptosis, there are also some features of inflammation, some features of apoptosis, and fever is also present. So this is pyroptosis, and it is also occur in some infection, and its details are also unknown, and its significance in human pathology is also unknown. Another mechanism of cellular death is autophagy. What is autophagy? Autophagy is basically a mechanism of cellular eating. When cell is deprived of nutrients, it starts eating its own constituents and its own organelles to obtain energy and nutrients. So this is mechanism is called autophagy. What happens when there are no nutrients? Endoplasmic reticulum of cell makes some vacuoles, which food vacuole. These food vacuoles are derived from its own cellular cytoplasm and own organelles, which are not crucial for cellular survival. So endoplasmic reticulum gathers some part of cytoplasm, some part of cellular organelles, and forms a food vacuole. This vacuole in turn fuses with the cellular lysosome and forms autophagolysosome. And this autophagolysosome, cellular constituents are digested and nutrients are absorbed for energy. And it is believed that this autophagy may have some association with the here is intestinal disease, which is inflammatory bowel disease, but the relationship is unclear, and it is not known well how autophagy plays role in inflammatory bowel disease. So here is diagrammatic representation of this disease. You can see these are cytoplasmic organelles, and this is formation of autophagic vacuole, and this vacuole is formed. This vacuole in turn fuses with the lysosome, and lysosome releases its enzyme and digests these components, and these nutrients and energy is used for cell to carry out its essential function. However, if autophagy cannot continue indefinitely, because as long as cell is consuming its own own organelles and own cytoplasm, with time these organelles will deplete, cytoplasm will deplete, and in the end cell will die out by apoptosis if nutrients availability is not made by some other way. So it was all about today's lecture, which we studied about apoptosis, necroptosis, pyroptosis, and autophagy. Thank you.